No, Andrew B. I don't know where Andrew D is. We're still missing him as well. Oh, there you are. All right. So we are going to talk about, we're going to continue talking about genetics. So for those of you who watched your videos and did your homework and were ready for your quiz today, you already know a lot of good information. So we're going to actually begin with the video I was going to show at the end of class yesterday. Just to review, because there's some terms we're going to use today, I want to make sure that you guys remember. They mentioned the term genotype. What is a genotype?
Gene combination for a trait.
Perfect. So the genotype is the gene combination for the trait. And we say it's made out of the alleles. So that's going to be the letters. Remember, we're using the alleles, our letters. And what's the phenotype?
The physical feature resulting from the phenotype.
The phenotype is going to be, yep, the physical manifestation or the physical feature. And it comes from or is derived from the genotype. We also heard in there, you have two different types of alleles. What are the two alleles?
Dominant.
So dominant and recessive. What can you tell me about the dominant allele?
Stronger of two genes expressed in hybrid represented with capital letters.
Yep, so this one here is going to mask the presence of a recessive if they're together. It's represented by a capital letter. And if something is a dominant trait, you need only one allele to have the dominant phenotype. That's why in the little video you just watched, if the mom gave them all dominant Y, and even though the dad gave them the little y, all the babies came out looking yellow like the mom. Because all you need is one dominant allele to have it. How about a recessive? What does that mean?
Gene that is masked by the presence of a dominant in a hybrid represented by a lowercase letter.
So gene masked by the dominant. You said it's represented by a lowercase letter. And you need two recessive alleles to have that trait. So again, in our little video we watched where it was little y, that's the only way you can get green. You had to have two little alleles or two lowercase alleles, recessive ones, in order for the recessive trait to manifest. We had talked about some other terms before as well. What does the term hybrid mean? What does the term hybrid mean? Hybrid means that it contains two different alleles. So what that means is it would have one dominant and one recessive. What was the other term for this? Yep, heterozygous. What was a purebred or true breeding? What did that mean? Yep, so it contains the same alleles. And it could be either homozygous dominant or homozygous recessive. Now, it's redundant to say homozygous recessive. Why?
because if it's recessive, it's already.
Yeah, in order to be recessive, you need 2 recessive alleles. So it's redundant. You know every recessive has to be homozygous. It's a really good point to remember. So we're going to talk about Mendel and his love for his pea plants. So he used the typical garden pea. It's actually the genus species name of it is Pisum sativum. And the reason he did this, you guys remember from your reading, is that it can be grown in a small area. If you watched the video last night, you saw how tiny his garden actually was. They produce lots of offspring. The offspring of a pea plant is the actual pea. And you know, peas come in pods, and there's more than one pea in a pod. So they have lots of offspring. They're also going to produce purebred plants when they're allowed to self-pollinate for several generations. And what did we say purebred meant? 2 of the same allele. So they're going to be homozygous. But he also used the pea plants because it was very easy to manipulate because he can artificially cross-pollinate them. And in case you don't remember what self-pollinated and cross-pollinated mean, we're going to talk about that in a second. In the event that you were wondering what a pea plant actually looked like, there is a pea plant right there. And you can see that it's got a very pretty flower on it. You've got the stems. Here is a pod here. This pod looks like it's probably what they would call smooth. In order to understand how Mendel did his work, you were required to understand a little bit about reproduction in flowering plants. And I know a lot of you have had this from middle school. So if we have a picture of the plant right here, these are the basic parts. You've got a stamen. And on top of the stamen, you're going to have anthers. And the stamen and the anthers, these are the male portions of the plant. The pistil is contained of the stigma, this part here, I'm going to change that color, is the style, and then the ovary. So the pistil is made out of the stigma, style, and the ovary. And these are the female parts of the flower. Now it's easy to remember that the stamen is the male part because it actually has the word men in it. So the stamen is your male part of the flower. The pistil, stigma, style, and ovary are your female portions. So the pollen of a plant actually contains sperm. It's the plant's sperm. And it's going to be produced by the stamen on those anthers. And these are going to be the male portion again. The ovary contains the eggs. And the ovary is contained in the flower of the plant for protective reasons. We've said before, how many of you enjoy eating apples? Those are the ripened ovary of an apple tree. Their eggs are now little embryos inside, and those are the seeds. How many of you are allergic to pollen? You're allergic to tree sperm. Congratulations. The pollen carries sperm to the eggs. And what do we call it when sperm and egg meet? For fertilization. Perfect. Bless you. We said that self-fertilization is when sperm and egg from same plant combine. Cross-fertilization, on the other hand, is where you have sperm and egg from different plants combine. And this cross-fertilization yields genetic variability. That's an important thing. So how did Mendel do this? And it's really quite ingenious. What Mendel did is that he hand pollinated all these different flowers using a paintbrush. And if you remember from the little cartoon we just watched about it, he had a paintbrush in his hand. It looked like he was cleaning the plants, but really what he was doing is collecting the pollen to go ahead and drop it down into the pistil. Doing that, once he got the plant pollinated, what he would do is he could snip off the stamens to ensure that no pollen from that plant got in there. So if you look here, here he is snipping, or that's not him, trust me, but that's someone else who's just removing the stamens to ensure, or the anthers to ensure, that no pollen is getting into that plant. And then to ensure like there was no cross-pollination based on like wind dispersal. If you remember from Food Inc., they were talking about how pollen from one cornfield could blow into another cornfield and then pollinate that corn, and they were then in debt to Monsanto. Well, he covered each flower with a cloth bag, like cheesecloth, to make sure that there was no contamination. So he was really, really sure about which plants pollinated which plant, and therefore he could kind of get a guesstimate of what their offspring would look like. He then traced the traits through several generations. So what Mendel was able to do was to produce pure strains, and we said pure strains are going to be homozygous, by allowing plants to self-pollinate for several generations. Now, even though it says that they were able to self-pollinate, he was the one who actually took their own pollen and put it inside that same plant. What are the actual traits and alleles that Mendel was studying? Well, there's a big old pile of them in your textbook. The first one that he looked at was seed shape. And your seeds, just like you saw in the little cartoon, they could either be round or wrinkled. What does that capital R tell you about the relationship between round and wrinkled?
Round is dominant.
Round is dominant to wrinkled. Very good. The seed color?
Yellow and green.
It could either be yellow, which is a capital Y, or it could be green, which is the lowercase y. So what does that tell you about the relationship between yellow peas and green peas?
Yellow dominant.
Yellow peas are dominant. However, when you eat peas, what color are they?
Green.
Green. So what does that tell you about the genotype of the peas that you eat?
They're both
It's homozygous recessive, or it's just little y, little y. The next one is pod shape. And it can either be smooth, is the way your book words it, or it could be wrinkled. You'll also see it written as inflated, but your book uses smooth, so that's what we're going to use. The pod color can either be green or yellow. The pod is the thing in which the peas are being carried in. Yes, Josh?
What's the line to the end?
Yes, it is. Because a big S and a little s look so similar, I always put a line through my lowercase so you can tell the difference. So for example, a big C and a little c look similar. I always put a line through my little c so you can tell the difference. When you are doing these Punnett squares and genetics problems, if I can't tell the difference between your capital letter and your lowercase letter, it's automatically wrong. So you need to make sure that you are extra careful with your sloppy handwriting. For seed coat, the seed coat is a little covering on the outside of the peas. It can either be gray or it could be white. Notice that I use G for both of those. If we were doing a dihybrid cross, we would have to differentiate better. However, for today, we're only doing monohybrid crosses, and the lowercase g and the capital G will suffice. For flower position, we said that the flowers, they could be axial, which would be capital A, or they could be terminal. Axial means on the stem or around the axes, whereas terminal refers to at the top or the terminus. For plant height, the plants could either be tall, which was dominant. Oops. Or they could be short or dwarf. And finally, flower color is purple, which is a capital P, which is dominant to white, which would be a lowercase p. Notice to show the difference between purple or the capital P and the lowercase p. The capital P is written in manuscript, whereas the lowercase p is written in cursive, so it has the tail on it to help differentiate those. It is of the utmost import that we can understand your writing. So just in case you're wondering what these things look like, the seed shape, here we are with smooth and wrinkled. Remember, smooth is dominant to wrinkled. Yellow is dominant to green in the color of the peas. There's our inflated or smooth and wrinkled. There's green and yellow pod color. We have our purple and white flowers. We have the axial and terminal. And then the tall and dwarf plants. Mendel went through and he did all of these plants. And remember we said, you don't remember if you didn't watch the video, that he grew like 28,000 plants, like an obscene number of plants. And when he did these, for example, with the trait of being smooth and wrinkled, he found that he got 296 to 1, or 5,474 would be smooth, which would be the dominant phenotype, and 1850 would be wrinkled, which is the recessive phenotype. And a comparison of the other characteristics as well, he was getting a consistent almost 3-to-1 ratio. So this 3-to-1 ratio here is very important. This here is going to be the phenotypic ratio of an F1 cross. What is an F1 cross? What does F1 mean? It's first filial generation, but it means it's 2 hybrids. And when I say it's 3 to 1, what that means is that you have 3 are going to show the dominant phenotype, and 1 will show the recessive phenotype. Based on Mendel's work, how did his work really measure up? Well, the theoretical or expected ratio of the plants producing round or wrinkled seeds, it should be about 3 to 1. Mendel actually observed 2.96 to 1. Any error in it is due to statistical analysis. Which means he's basically right. It's basically the same as the theoretical yield. However, the larger the sample size, the more nearly the results will mimic the theoretical. What this means is that if you do a Punnett square based on your genotypes, it doesn't mean that if you have 4 kids, 1 child would get the recessive and 3 will have the dominant. It means every single time you have a child, there's a 25% chance of having the recessive phenotype and a 75% chance of having the dominant phenotype. Bless you. My goodness, are you okay? Okay. So what is the P1 generation? What did that mean? This is the parental generation. What is their genotype always? They're going to be homozygous for contrasting traits. What does that mean?
One is homozygous with the dominant and one is homozygous with the recessive.
Perfect. So one is going to be homozygous dominant and one of them is going to be homozygous recessive. That's what that means. The letters don't mean, that it's only specific for this R trait. It's saying any trait. Homozygous for contrasting traits means that you have 1 homozygous dominant times 1 homozygous recessive every single time for a P1 generation. What is the F1 generation? It does mean first filial. This is going to be our first offspring in a breeding experiment. It does mean first filial. And it comes from breeding the P1 generation. That's from our parent generation. So the offspring of the parents are going to be the F1 generation. This F1 generation are going to be all heterozygous. And that's when I say heterozygous, am I referring to their genotype or their phenotype? Genotype. Their phenotype would be that they all look dominant. What's the F2 generation? So the second generation offspring. And this comes from breeding the F1 individuals. So if we take a look at an example here, the trait we're going to look at is going to be plant height. So our P1 generation now for the fun part, we actually have to do some. So we're going to do the monohybrid cross. What does mono mean? One. So this is going to be one trait. We're going to only look at one trait at a time. Let's do a P1 monohybrid cross together. The way in which I'm going to do this should show you how I expect you to do it from now on in class or on your homework. The first thing we have to do is determine what our cross is, and you have to let your readers know that. So what are the things that are in a P1 cross? It's always going to be the same. One homozygous dominant and one recessive. So what trait do you guys want to do? You can choose any of the ones that Mendel has done. We just did that. You want to do smooth and wrinkled? That's what the other class did too. I think it's odd. So the first thing I'm going to write is my key. So capital S we said was smooth because that was dominant. And my lowercase s is equal to wrinkled. So when you do a Punnett square, you're always going to want to include a key that lets you know what the alleles stand for. If you don't have a key, it's wrong. Now we need to show the cross. What is the cross between? For a P1, you told me it's homozygous dominant, so what would that look like? What would the genotype be there? Capital S, capital S. And a recessive, which would be what? They're all S's. You have to be two lowercase s's. You also need that. That's actually showing the cross you're performing. So besides having the Punnett square filled in, you need to have a key and show the cross. Now your next step is to go ahead and fill the alleles around the outside. Remember, this is your diploid number up there, the capital S, capital S. We need to do gamete formation and turn it into the haploid. So we're going to have big S here and a big S here, and then little s and little s. The next step is just to fill it in. They're going to combine. And what process is this that we're showing in each box? What?
Fertilization.
It's fertilization. Very good. So you've got gametogenesis, or the forming of gametes on the outside, and you've got fertilization shown in the middle, or in each box. What are our possible genotypes here? What's the possible genotype?
Capital S, lowercase s.
Big S, little s. Very good. What's our possible phenotype?
Smooth.
They're all smooth. You know it. What's our genotypic ratio? Let's use a percentage here because it's easier. 100% what? You have to tell me what. Nah, that's not a genotype. All of them are all uppercase S's? 100% big S, little s. Very good. How about the phenotype? Now it's 100% smooth. Notice when I'm writing out my ratio or my percentage, I'm telling you what they are. It's 100%. 100% of what? 100% smooth. 100% big S, little s. So to review it, remember a monohybrid cross is going to be a homozygous dominant times a homozygous recessive. What are all of our offspring? What do they all look like? They're all going to show the dominant phenotype. How about their offspring? What do we call those? What? They're the F1. But what do they look like? Or, I'm sorry, what is their genotype going to be? They're all going to be hybrids or heterozygotes. The genotypic ratio you told me is going to be 100% heterozygous. And the phenotypic ratio is going to be 100% dominant phenotype. Now let's take our results of the P1 generation and let's make an F1 cross. So what are the first things I have to do before I can start doing a cross?
Make it key.
I got to make my key. So we're going to carry that through. We're going to still have big S equals smooth. Little s equals wrinkled. And now I've got to write out what my actual cross is. In an F1 cross, remember we take the offspring of the P1 generation. What is my F1 cross going to look like?
Uppercase S, lowercase, uppercase, lowercase.
Like this?
Times.
Oh, okay. So the times in there is what indicates that it's a cross. Now what's my next step? I have to determine my gametes by splitting up my alleles. So I'm going to have big S, little s here, big S, little s here. And then my next step is fertilization. Big S, big S, big S, little s, big S, little s, little s, little s. What are our possible genotypes here?
Big S, big S, big S, little s, little s, little s.
So I have 3 different possible genotypes. What are my possible phenotypes? Well, let's figure that out. What is this first box going to look like? That's definitely smooth. How about this one? How did you know that one was smooth? It has 1 dominant allele. This one here is the same as the other one, so that's going to be smooth. And how about this one? Wrinkled. So my phenotype is going to be what? I have smooth and wrinkled. What's our genotypic ratio going to be? In this case, I'm not going to use percentages. I'm just going to use numbers. My genotypic ratio, I have 1 to 2 to 1. Now, when you write out a genotypic and phenotypic ratio, you need to indicate, again, what those genotypes or phenotypes are. So when I write it, I always write the numbers on the top and then underneath it, what they are. So that means I have 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. How about our phenotypic ratio? What is that going to be? 3 to 1, that golden ratio that Mendel got. And it's going to be 3 are going to be smooth to wrinkled. If you don't indicate what your options are, it's going to be wrong. So remember, an F1 is always going to be heterozygote times a heterozygote, or 2 hybrids that are going to be combining. So the offspring, what percentage of the offspring are homozygous dominant? Well, we had homozygous dominant. That means that are going to have 2 capital letters. We had 1 out of 4, which is what? 25%. How many of them, what percentage are going to be heterozygous? 2 out of 4 we did, right? So it would be 50%. And how many are going to be homozygous recessive? 1 out of 4 again, which would be 25%. What is it called? What do we call the offspring of the F1? The F2 generation. What is the genotypic ratio going to be again? 1 to 2 to 1, and it's always going to be 1 homozygous dominant to 2 heterozygous to 1 recessive. Always. What about your phenotypic ratio? It's going to be 3 to 1. It's always going to be 3 exhibit the dominant phenotype, whereas 1 exhibits the recessive phenotype. I don't think class is over yet, right? No.
Yeah, it is. It's Thursday.